I want to broaden the conversation a bit beyond just sun exposure and talk about a more general principle that UV damage illustrates particularly well: the idea that environmental stressors can create lasting changes in how cells function, changes that persist even after the stressor is removed. This concept is important because it helps explain why past sun exposure continues to matter for future cancer risk. Scientists call this phenomenon stress memory or stress priming. Cells that have experienced significant stress can be altered in ways that change how they respond to future challenges. Think of it like this: a cell that's been through a traumatic event doesn't just return to its previous state once the trauma passes. It carries forward molecular changes that reflect that history. Some of these changes happen through epigenetic mechanisms, chemical modifications to DNA or the proteins around DNA that affect gene expression without changing the genetic sequence itself. UV exposure can add methyl groups to certain spots on DNA or alter histone proteins, and these modifications can persist through multiple rounds of cell division. The cell's DNA sequence might be intact, but the pattern of which genes are turned on and off has been permanently altered. There's also something called cellular senescence that I mentioned earlier, and it's worth exploring more deeply in this context. Senescent cells are cells that have stopped dividing in response to stress or damage. They're not dead, but they're not actively participating in tissue function either. And crucially, senescent cells secrete inflammatory molecules, something called the senescence-associated secretory phenotype. These cells are essentially broadcasting stress signals to their neighbors long after the initial stressor is gone. As senescent cells accumulate in tissues and UV exposure accelerates their accumulation, they create a persistently inflammatory environment. This affects how neighboring cells behave, can impair tissue repair, and can create conditions favorable to cancer development. So even years after significant sun exposure, the presence of senescent cells in your skin is maintaining an altered cellular environment. Research has also shown that stress can alter mitochondrial function. Those are the cellular power plants that generate energy. UV-damaged cells often show persistent changes in how their mitochondria work, producing more reactive oxygen species and less efficiently generating energy. These mitochondrial changes can affect overall cell health and stress resilience for the cell's remaining lifespan. What's particularly concerning is that some of these stress memory effects can make cells more vulnerable to future insults. A cell that's been UV damaged once might respond more severely to subsequent UV exposure or to other stressors. It's like the cell's stress response has been sensitized, making it overreact to challenges that a stress-naive cell would handle easily. But here's a more encouraging angle on all this. Understanding stress memory has opened up potential intervention strategies. If we can identify the molecular marks of stress memory, we might be able to reverse them or prevent them from forming. There's research into compounds that can clear senescent cells, for example, or therapies that can reset epigenetic marks. These are early-stage investigations, but they suggest that even lasting changes from past stress might not be completely permanent. More practically, the stress memory concept reinforces why ongoing protection matters. Every new stress event potentially compounds the memory of past stress. But conversely, extended periods without additional stress might allow cells to gradually return toward normal function, to clear out senescent cells through natural turnover, to reset some of the epigenetic marks that were altered. Protection isn't just about preventing new damage, it's also about giving your cells the most favorable conditions to recover from old damage.